Harvesting and processing of berries. Maceration of berries and obtaining botanical seeds. Approximately 48 days following pollination, if the berries are fully developed, harvest them in kraft paper bags, duly identified with the name or the code of the progenitors that participated in the process, and the date and the number of the berries harvested. Then, leave the berries to mature inside the bag, in the environment, until they are soft and give off a particular odor, sign that they are ready to continue the process. Before beginning the seed extraction, place a parchment paper bag inside each kraft paper bag with the aforesaid identification information and the date of the extraction of the seeds. Following that, fill a pitcher with running water. One by one, squeeze out the berries immersed in water to release the seeds, process known as maceration. Upon completion of the maceration, remove some water and pass all the contents through a 25 mesh coarse net, which will prevent the remains of the carpels from passing through with the seeds. Thus, only the seeds will be received in another pitcher. Very carefully, rinse the seeds with water several times, making sure that they are not trapped in the remains of the net. Then, collect the seeds by pouring the contents of the latter pitcher in a thin piece of a 50 mesh net, previously folded in 4, forming a funnel to receive the seeds. Rinse the pitcher again with running water and pour it back into the net to retrieve all the seeds. Then, wash the seeds under running water to remove any traces of mucilage and rinse the net externally to completely clean it. Gather the seeds using a thin trowel. In this case, we're using a plastic stake, and place them in the corresponding parchment paper bag. Once closed, place the bags on a drying rack to allow them to dry at ambient temperature for approximately 6 hours. Cleaning, counting, dehumidifying, packaging, and final storage of the seeds. Once the seeds are dry, clean and count them in the laboratory. Previously, wash and disinfect your hands. Pour the seeds onto a piece of paper and, using a stylus previously disinfected with alcohol, separate the seeds that are blackened, empty, or without an embryo, while counting the visually healthy ones. If the seeds are abundant, an estimate of the total can be made with the help of a small test tube. Using a fine point indelible marker, mark the height reached by the volume of 200 healthy seeds. Then, using the test tube as the measure, calculate the amount of the selected seeds in the sample. After that, transfer the selected seeds, using a small funnel, to an aluminum foil envelope for its final drying. The envelope must display the code number of the progenitors, the year of crossing, the number of seeds, and the code assigned to the family, if any. Dry the seeds in a desiccator cabinet for as long as necessary, until the device registers a relative humidity of 4 to 5%, which can take from several days to several weeks. Otherwise, if a desiccator cabinet is not available, a container with silica gel in the bottom can be used. Upon completion of the drying time, remove the envelopes from the desiccator cabinet and seal them using a bag sealing machine. Afterwards, place the envelopes one by one in a larger aluminum foil envelope, which must display the same information written on the small envelope. If a barcode system is available, a self-sticking label containing the same information can be additionally placed. Finally, seal the envelopes with a bag sealing machine and transport them on a tray to their final storage destination in a cold room or refrigerator at 4 degrees Celsius. Seed viability test. Under the indicated storage conditions, the seeds will have a dormancy period of 6 to 24 months. Therefore, after 6 months, germination testing for the families projected for sowing can be initiated. For this purpose, place a sample of 100 seeds of each family in small plastic containers and take them to a humidity chamber for 24 hours. Then, take the samples out of the chamber and separate 2 replications of 50 seeds each. Place each one on filter paper into a properly identified petri dish of 9 centimeters. After that, moisten the filter paper by adding 5 milliliters of distilled water. Finally, incubate the petri dishes in a germination chamber or incubator at 27 degrees Celsius for 8 days. The emergence of the radicle is a criterion used to determine whether a seed has germinated. Results are expressed as a percentage of seeds germinated or viability percentage. Start the germinated seed count on the 3rd day, when the seeds begin to germinate, and perform it daily until the 8th day. Remove the germinated seeds To avoid confusion with the ones not germinated yet, always keep the filter paper moist by adding distilled water when necessary. A germination percentage above 50 within 8 days indicates that the seeds of that family have broken dormancy and therefore are ready to be used immediately. Otherwise, place a sample in the incubator for additional 7 days at 17 degrees Celsius. If after this treatment the germination percentage is above 50, it is interpreted that the seeds of the family from which the sample was drawn were dormant. In case the sample of the seeds does not germinate after this second treatment, the seeds of that family are considered of poor quality and should be discarded. If you desire to accelerate the dormancy break of certain family seeds, you can store them from the beginning at 30 degrees Celsius at a moisture content below 5% for a period of 4 to 6 months. After this, take a sample for a germination test. If the seeds are still dormant, extend storage under the same conditions, testing for germination every 2 or 3 months until dormancy has been broken.